Hi, everybody. Welcome to this webinar. It's a pleasure to announce to you that we have beautiful speakers on an important subject. My name is Erika van den Akker. I'm a pediatric endocrinologist from Erasmus MC Rotterdam and coordinator of the pediatric part of our obesity center CGG. I will be moderating and chairing this session and will be introducing the speakers in a minute. But before I do that, I would like to start with saying that we are honored and glad that this webinar was organized by a joint mission of 3 organizations. That is the European Society of Endocrinology, together with the Endo-ERN and ESPE. So it means that we will cover both pediatric and adult subjects on genetic obesity disorders today. Why is this topic important? Well, genetic obesity disorders is a field that is developing fast. Every year there are novel genes discovered, new diagnostic techniques, and there is also new possibilities for treatment. So therefore, it is becoming more and more important to identify these patients and to give them the right treatment. So it means that we will start today. Sorry, before I start, there are still some practical things to mention to you. First, I would like to say that we want to thank one of the sponsors for this webinar, which is Rhythm Pharmaceuticals. And it is important to stress that the speakers develop their presentations completely independent of that. If you have questions please just put them in the Q&A box. You can do that even during the presentations, and we will cover them after. There will be a little bit of time immediately after the presentation and at the end of the webinar. So please don't put them in the chat, but put them in the Q&A box. This webinar will be recorded, and after 3 weeks, it can be looked back following the ESE website. So that's the practical stuff for now. So let's go immediately to the 2 wonderful speakers. The first speaker is Dr. Peter Kuehnen, my colleague from Berlin, Charité. Peter, if you want to start, the floor is yours.
Thank you, Erika, for this kind introduction. And of course, I would like to thank the organizers for this kind invitation. So ESPE, ESE, and Endo-ERN. And in the next approximately 15 minutes, I would like to give you a brief overview about the new findings which have been identified in regards to genetic obesity, polygenic risk scores, new monogenic forms of obesity, and I would like to pinpoint a few information about the gene-environment interaction. And this should set the stage for the next talk of Elizabeth, who will continue along this line and will talk about examples and their results in their cohorts. These are my disclosures. I would like to stress that none of these disclosures affect or influence the content of this talk. And I would introduce the overall topic with this color-coded map. And what you can see here is the BMI the mean BMI in each country. And this is the situation 1970. And you all know that in the last 30 years, the obesity prevalence has tripled. And not only industrial, but also in industrial and low and middle income countries. But the importance of this topic becomes much more obvious if you analyze statistics why people die these days. And some of you will have read this publication in the New England Journal of Medicine in October this year. And here, large consortium analyzed the impact of 5 modifier risk factors. So obesity, high blood pressure, high cholesterol, smoking, and diabetes on the risk to die due to a cardiovascular event or death because of any cause. And it becomes obvious that over 50% of deaths related to cardiovascular events are related to this modifier risk factors, and approximately 20% of any death is related to these risk factors. And if you separate each of these risk factors, it turns out that obesity is relevant for approximately 7% of deaths in relation to a cardiovascular event. And this fits nicely with recent statistics. And this is the Global Burden of Disease report. And again, here, and this was 2017, again, one of the most important risk factors for the deaths in industrial countries is high blood pressure, and then high blood glucose levels are on the third place, and the fifth is obesity. And if we address that obesity is the most important risk factor for high blood glucose levels and for hypertension, it becomes clearly evident that we need to change our way of treating obesity, and that we need to understand the underlying mechanisms for the individual risk to develop obesity, because if we find a solution, or if we might be able to improve the situation for the patients, it might be really, really relevant for the individual, but also for the healthcare system. And we know since decades that, on the other hand, our body weight is quite strictly regulated. And one of the best examples and one of the fundaments for this hypothesis were based on studies with monozygotic twin pairs. And this here is a meta-analysis. And in the study, they analyzed the BMI concordance in different cohorts with monozygotic twins. And on the x-axis, the number of each dot represents a study, and on the x-axis, the number of included twin pairs is reported, and and here the BMI concordance is shown. And it became clearly evident that monozygotic twins are really, really similar regarding the body weight, also regarding the height, but also regarding the body weight. And so you might say that this is quite logical, because most of the monozygotic twins might grow up in the very same place, in the very same environment. But if the monozygotic twins are separated within the first month of their life, so one is living then afterwards in the United States, and the other twin is living in Europe and is growing up in Europe, still the high BMI concordance persists. So there is a strong regulatory moment regarding BMI. And then maybe some of you have seen it as well in Science in September this year. There was a very nice report about the relevance of genetics, and they pronounced and explained one misunderstanding of these data, because several people were arguing that in the last 30 years, genetics haven't changed. Still, we have this high rate and high increase of obesity prevalence. So that the majority of patients with obesity might not be related to any genetics, but might be more related to the change of environment and the less of activity, which is now present in our daily life. But we need to confess that the underlying mechanism is a medal with two sides. So on the one hand, we have the genetic or epigenetic regulation, and on the other hand, we have the environment. And there's a very strong interaction between both sides of the medal. The last 10 to 15 years, a lot of money and a lot of effort were put into the investigation of any genetic causes for obesity. And large genome-wide association studies have been performed. And this is most of the time presented with this Manhattan blots, and several genetic variants have been identified to correlate with body weight. And one of the most famous genetic variants has been identified near the FTO gene. But in general, all these data from the genome-wide oscillation studies explain less than 15% of inter-individual body weight variability. But then groups started to study thousands, hundred thousands of genetic variants in the very same time in large cohorts of millions of people. And Lee Kaplan and his group were the first ones who were able to calculate the polygenic risk scores. And what they have done in really large cohorts was to describe the genetic profile of individuals with a low body weight, normal body weight, and high body weight. And then they calculated each genetic signature, and then they addressed this, and by analyzing independent cohorts, for example, in this case, a large cohort with individuals with severe obesity, and then they investigated whether this high risk genetic profile might be more common in this group. And it turned out, and this is shown with this dark blue line, that in this obesity group, carriers of a genetic profile which might lead to increased risk to develop obesity were much more common than carriers of a genetic signature which has been identified originally in lean individuals. So what does it mean? We could speculate that in your town, maybe 100 babies might have been born today. And some of them might have a genetic profile which is associated in these studies with a high risk to develop obesity. And a large number of babies might have a genetic signature which is not associated with obesity. But all these babies with a high risk profile, all of them won't develop obesity later in time. So just 4 out of 10. And also these babies with a genetic profile which is correlating with normal body weight, all of them won't stay lean. Some of them will develop obesity. And so we need to confess that if you have a high genetic risk score for obesity, your risk to develop obesity is 4-fold increased. But it's not 100%. And so in the end, at the moment, polygenic risk scores, or with polygenic risk scores, you might classify the individual in 4 out of 21 cases correctly, and in the other cases, it is wrong. So there is a strong genetic regulation. There are genetic variants which are associated with obesity, but we are far away from having classified and clarified this. And because there are additional factors, and as I said, the environment plays a role. And this is a study from a Scandinavian group, which is somehow underestimated, because this group in Scandinavia, they analyzed polygenic risk scores in individuals living in the 60s and compared this with the polygenic risk scores living in the around the year 2000. And then they calculated what the impact of the polygenic risk scores regarding the BMI. And it turned out that it was different between men and women. And in the 60s, high polygenic risk scores predisposed to approximately 1.2 BMI points higher BMI than compared to the least predisposed. And in around the 2000, this polygenic risk scores was associated with 2 BMI points increase compared to the least predisposed individuals. So it's still in the 60s, there was an impact of this polygenic risk scores, and it's becoming even more intense if the environment is changing. So there's a gene-environment interaction. That specific genes are highly important has been identified decades ago in the 1990s. And this mouse was the starting point, somehow the OB mouse. It was somehow a revolution when Jeff Friedman and Reed Leibel identified the genetic cause why this mouse model is becoming obese and has such a strong hunger feeling. And at this time, they identified that also hormones and some genes are involved in our body weight regulation and could lead to the development of obesity. And they and other groups identified the so-called leptin-melanocortin signaling cascade, and in which there are hormones like ghrelin or insulin, but one of the most important hormones is leptin. And leptin is activating leptin receptors in a key region in the hypothalamus. And leptin regulates two different neuronal populations within the hypothalamus and the Akker nucleus. On the one hand, they are neurons which are expressing agouti-related peptides, and on the other hand, they express POMC. And the activation of POMC neurons is leading to the secretion and production of a second hormone, which is called melanocyte-stimulating hormone, alpha and beta MSH. And this is a ligand for G protein-coupled receptor, the melanocortin-4 receptor. And on the other hand, these neurons which are expressing agouti-related peptides are inhibited by the activation of the leptin receptor, and agouti-related peptide is the inverse agonist of the MC4 receptor. So both neural populations are regulating centers, and in this case, they are regulating our feeling of satiety and thereby our body weight. And if they are genetic variants, so mutation in one of these genes of the leptin-melanocortin pathway, so leptin receptors, pro-melanocortin, POMC, or the MC4 receptor, this is leading to a disruption of pathway function and to a severe feeling of hunger, so called hyperphagia. And this is true for rodent models and also for human beings. And this is one typical weight course of one patient with monogenic obesity. This is the age of the patient until this patient became an adult. And you can see easily that the hyperphagia led to an early onset type of obesity. So the obesity started before the age of 5. And although the patient and the family tried hard to control body weight, this feeling of hyperphagia was so strong that there was a continuous weight gain. And so conservative treatment options weren't successful to reduce body weight. And that's why in several cases, the impact of bariatric surgery is discussed in cases with monogenic obesity. And once we summarized a few patients with monogenic causes, all variants were in one of these genes related to the leptin-melanocortin pathway, either in the leptin receptor gene, POMC gene, or MC4 receptor gene, and they received bariatric bypass operation. And you can see easily that all of them had an early onset of obesity, and with the surgery, there was a short period of weight reduction in all cases, but then due to the fact that this operation is not leading to any regulation or any change of this feeling of hyperphagia, the weight started to increase again. And we are overlooking now a couple of patients. There are some patients which responded to bariatric surgery a little bit, but the vast majority is not responding to bariatric surgery. So we need to be very cautious who will be operated. And that's one reason why it matters to identify these patients. And the other point is that now we have a few treatment options for these patients. And this is the very first example when Jeff Friedman treated the leptin deficient mouse with leptin, because this led to fundamental change of the food intake, because a feeling of satiety has been activated, and thereby this led to clear reduction, normalization of body weight. And leptin is could be used to treat leptin deficient patients, also humans, and for patients with leptin receptor deficiency or POMC deficiency, an MC4 receptor agonist, setmelanotide, has now been approved, and this is leading also in these patients to reduction of the hyperphagia and body weight. But I would like now to focus a little bit on 4 different genes where we where the community identified new findings and were able to improve a little bit our knowledge about the regulation of the satiety center. And here I would like to concentrate your attention on the melanocortin-4 receptor. Because if you perform this large genetic tests and analyze huge cohorts with individuals with obesity, without obesity, the region, the gene where you will identify the most potent genetic variants which explain the largest amount of body weight variability will be the MC4 receptor. And this has been shown in several studies. And not biallelic, but heterozygous variants within this gene are really common. And this is a study performed by Stephen O'Rahilly and Giles Yeo in Cambridge. They analyzed a cohort longitudinally and analyzed the MC4 receptor gene, and then they compared the weight courses. And then they identified variants which were behaving like the wild type. Then there were variants which led to partial loss of function, and some variants led to complete loss of function of this gene. And I would like to focus the attention on this dark blue cycles. These are variant carriers with a complete loss of function variant, and step by step, approximately starting with the age of 5 years, they started to have an increased weight, not 50 kilos, 100 kilos, but 10 to 20 kilograms. So here, the heterozygous variants seems to be a very strong genetic risk factor. They are not the single cause for obesity in this case, but they are a very strong risk factor. And the prevalence was really high, so 0.3%. So somehow it matters to identify these patients because they have the strong risk factor, and there might be other reasons why they might be predisposed to develop obesity. And in this case, it might be a little bit more difficult to treat them because weight loss might not be so easy. And so in general, if we focus on these patients, it is relevant, and Elizabeth will pinpoint a little bit the red flags where genetic testing might make sense. On the one hand, variants will be identified which are related to the leptin melanocortin pathway, and most of these variant carriers will have a normal development, normal cognitive development, but might have some endocrine features. On the other hand, there are patients which have, in addition to obesity, some remarkable findings in regards to their cognitive development. So there might be a developmental delay, or they might have learning or behavioral difficulties. And this is also a group where genetic testing makes sense, because there are some diseases like celiopathies, like Bardet-Biedl syndrome, in which the genetic defect explains a particular spectrum of phenotypic abnormalities like retinal dystrophies or skeletal abnormalities and obesity. Then there are genetic defects which are leading to obesity and symptoms which are related to the autism spectrum disorders, and some have behavioral abnormalities, anxiety, aggression, or hyperactivity. And also here there are genetic defects which might explain these phenotypic spectrum. And there's recently the group of Akker in Leipzig identified a new variants in a gene, and this is related to this mouse model. Some of you will know this because this mouse model is known since decades. It's a agouti, viable yellow mice. And in this rodent, the agouti gene, which I introduced a few seconds ago in the leptin melanocortin pathway, agouti is the inverse agonist of the melanocortin-4 receptor. In this case, a genetic variant in this rodent is leading to an overexpression of this agouti protein, and this is leading to severe hunger feeling, and it affects also the fur color, and that's why this rodent has this yellow fur color and is hyperphagic compared to the wild type mice, which has a brown fur color and is not obese. And the group in Leipzig, they analyzed, first of all, a patient with obesity. This patient had bariatric surgery, which led to reduction of BMI and then to BMI increase again. This is the height of the patient. So this patient was a little bit taller. And in this case, they observed a genetic variant, an alteration in the gene called ASEP. ASEP stands for agouti signaling protein, and it is an inverse agonist of melanocortin receptors like the MC4 receptor. And this led, this variant led, and this is very similar to this rodent model, to an overexpression of this protein, and then this protein could influence and have an impact on the MC4 receptor and might explain hyperphagia in this patient. And it affects also the MC1 receptor, which is expressed in the skin and which is relevant for the production of eumelanin. And this might be one reason why this girl has such a red hair color. And the genetic variant is somewhat peculiar and interesting because it's a tandem duplication. This is shown here. This is the ASEP gene, and this is followed downstream. There's a gene called itch. And interestingly, there's a breakpoint which is leading to the duplication. And this duplication here is leading to the situation that the ASEP gene is becoming under the control of this itch promoter. So now the itch promoter, which is expressed ubiquitously in different tissues, is now leading or is driving now ASEP expression. And this is leading to this overexpression. And interestingly, this breakpoint and this duplication has been identified years ago also in other animals like the sheep, for example. So there might be something very interesting which is related to the evolution and which is related to this genetic environment. And the group performed functional characterization of ASEP expression, and they observed, for example, in peripheral blood cells, where ASEP is not that much expressed, that there's a huge overexpression in the patients. So this is one new gene, and the second gene in which new genetic variants have been identified in patients with obesity is the serotonin 2C receptor. This has been published by the group in Cambridge from the group of Sadat Faruqi, and they identified a couple of mutations in the serotonin 2C receptor. It is a G protein coupled receptor, and this led to dysregulation of our hunger feeling or our body weight. So they had all history of obesity, but also to changes of the behavior. And finally, I would like to stress that there is a third genetic region, which is quite interesting. And this genetic region is coding for the GS protein of GPCR. So this is, as you know, there are several G protein coupled receptors which are recruiting different G proteins, so GQ, GI, etc., but also GS, and the recruitment is leading to an activation of CNP within the cell. And the melanocortin 4 receptor is one of the receptors which is coupling to GS, also to other G proteins like GQ, but also to GS. And if there is a genetic variant in this GS gene, this signaling might be impaired. And there were two publications where it has been shown that carriers of genetic variants within this GS gene, it's called the GNAS locus, is associated with an increased BMI and increased body weight. So in summary, I hope I could present you the recent findings about the polygenic risk scores, the relevance of common genetic variants in interaction with the environment for the development of obesity. I presented showed you some data about the relevance of the leptin melanocortin signaling cascade, that there are biallelic variants within one of these genes, so leptin receptor POMC, are leading to severe hyperphagia and early onset obesity, which persists until adulthood. And then there are new findings and the identification of new genetic variants in the ASEP gene, in the GNAS gene, in the serotonin receptor. And the more common genetic testing will be in the future, the more I would expect that there will be a couple of more gene regions where genetic variants are leading to the obesity development. And with this, I would like to thank the whole team, and of course, for you for your attention. Thank you so much.